A shadow glides across the shallow reef, and within a quarter of a second, it completely vanishes. We call it camouflage. We assume it is an automatic response, as mechanical and reflexive as a chameleon changing skin. But in June of 2023, researchers at the Okinawa Institute of Science and Technology slowed the footage down to individual frames, and they discovered a signature of mind we never expected. An overshoot, a brief pause, and a correction. Stay for the 32-lobe brain atlas reveal. There's a specific number of lobes that reframes the whole story. To understand the strange nature of this discovery, we must look at the animal everyone compares the cuttlefish to. Chameleons change color through light-activated pigments, a process that happens at the surface of the skin. It requires no active neurological debate. The skin simply reacts to the ambient light, shifting colors automatically. This is a passive biological reflex. The brain is not deciding what color to become. But the cuttlefish is fundamentally different. Its disguise is not a reaction to light. It is an active muscular performance directed by the central nervous system. In the cuttlefish, every single spot of color is controlled by a dedicated organ. These organs are called chromatophores, millions of tiny pigment sacs embedded in the elastic dermis. Unlike a chameleon, these sacs do not rely on slow chemical transitions to change shade. Instead, each chromatophore is surrounded by a ring of microscopic radial muscles. When these muscles contract, they pull the pigment sac open like a miniature parachute, expanding the color. When they relax, the sac shrinks back into a tiny, near-invisible dot. Because every muscle ring is directly wired to a motor neuron, the brain has direct control. The nervous system is pulling thousands of individual muscular triggers every single second. This is Insight 1. In their landmark work, Hanlon and Messenger proved that cuttlefish camouflage is a system of motor control, not slow light physics. This neurological wiring raises a profound question. How does a single brain coordinate this massive orchestra of color? To find out, we have to look inside the physical brain of the cuttlefish itself. Until recently, the internal mapping of this brain was largely a mystery. We knew the brain was large for an invertebrate, but we did not know how it was organized. Now, let us pause. In 2023, a breakthrough changed everything we knew about their cognitive architecture. A collaborative team at Columbia University's Zuckerman Institute spent years mapping the cuttlefish brain. Using high-resolution MRI scans, histology, and deep learning, they created the first complete brain atlas. They mapped a level of structural specialization that rivals many complex mammals. They discovered that this brain is divided into 32 distinct functional lobes. This is Insight 2. The Shomrat study mapped the 32 brain lobes of Sepia bandensis, showing that the optic lobes occupy 3/4 of the entire brain volume. This massive visual processing center sits directly adjacent to the chromatophore control lobes, and right next to that sits the vertical lobe, the area responsible for learning and long-term memory. This physical layout hints at something incredible. Memory and motor control are tightly coupled. The cuttlefish is not just seeing and reacting, it is referencing memory to hide. Before we explore how this memory works, I have a quick request. If you enjoy these deep dives into animal intelligence, please subscribe. WildMind releases new episodes every Wednesday and Saturday. Now, let us look at the behavioral evidence that proves this brain is calculating, not reacting. When a cuttlefish transitions to a new background, we see it change instantly. But high-speed video analysis shows the process is much more complex. The animal does not just transition from pattern A to pattern B in one smooth step. Instead, it runs an active calibration process that researchers only recently managed to decode. They discovered that the cuttlefish routinely overshoots its target pattern. It's like a designer hitting Ctrl-Z on Photoshop, adjusting opacity frame by frame until the fit is perfect. This temporary overshoot is a classic signature of a feedback loop. It shows the brain is actively comparing the skin's output to the surrounding environment. The brain reads the error, calculates the difference, and adjusts the muscular tension. This is Insight 3. The 2023 Sachse and Deco study in Nature proved that cuttlefish camouflage is an iterative, feedback-driven calibration, not a reflex. This feedback loop operates on a timescale of milliseconds, yet it involves active visual monitoring and continuous physical correction. This is identical to how a human guides a hand to grab an object. You do not just throw your hand and hope. Your brain monitors the distance and adjusts. The cuttlefish is doing the same with its entire body surface. This calibration suggests that the disguise is driven by internal state, not just external cues. If camouflage were a simple reflex, the output would only change when the background changes. But researchers began to notice that the skin's dynamics fluctuate even in static environments. The pattern is not static, it breathes with a subtle rhythm. This rhythm turns out to be a direct reflection of the animal's internal physiology. In 2024, scientists decided to test how internal physiological states affect this skin rhythm. Now pay attention. They discovered that a basic bodily need completely rewires the skin's rhythm. They placed hungry and well-fed cuttlefish on identical backgrounds and recorded their skin. The visual inputs were exactly the same, but the skin output was completely different. The hungry cuttlefish produced patterns that fluctuated at a much higher temporal frequency. Their skin was physically more active, reflecting a state of heightened arousal. The well-fed cuttlefish were calm, their skin dynamics exhibiting slow, stable rhythms. This is Insight 4. Cohen-Bodenes and Neri proved that internal physiological states like hunger modulate skin pattern dynamics independently of visual inputs. This discovery breaks the traditional model of camouflage as a simple copier. The cuttlefish skin is not a photocopy machine. It is a projector of its internal state. It is comparable to human breathing. It happens automatically, but is modulated by stress or hunger. This indicates that the decision to camouflage is a negotiation between internal and external worlds. To make this negotiation work, the brain must run two distinct visual systems simultaneously. These two systems process different aspects of the environment at the same time. The first system looks at the local visual texture directly underneath the animal's belly. It maps the fine details of the sand, rocks, or algae it is resting on. This requires high-resolution local mapping to ensure a perfect micromatch. The second system maps the global statistics of the entire background. It calculates the overall color temperature, average brightness, and dominant shapes. This ensures the animal blends into the larger landscape, not just a single rock. But what happens when these two systems disagree? When local and global visual details conflict? This occurs when a cuttlefish is sitting on fine sand, but surrounded by large dark rocks. If it matches the sand, it stands out to predators looking from a distance. If it matches the rocks, it stands out against the white sand directly beneath it. This is a classic decision-making conflict. The brain must resolve it in real time. Watch closely. Researchers found that this conflict triggers a live neural negotiation. The brain does not choose one system and ignore the other. It runs a negotiation. The overshoot and correct pattern we saw earlier is actually this negotiation in action. The brain is testing compromises, checking the visual feedback, and adjusting the muscular tension. It is an active computational debate between different parts of the 32-lobe brain. This negotiation ensures the final pattern is the safest possible compromise. This is Insight 5. Chung and Marshall proved that cuttlefish combine local visual texture and global background statistics, resolving conflicts through real-time neural negotiation. This cognitive complexity becomes even more shocking when we look at the cuttlefish's eyes. Cuttlefish are famous for their ability to match the exact colors of the reef. They can match reds, greens, and yellows with photographic accuracy. Yet, from a physiological standpoint, cuttlefish are completely colorblind. They possess only a single type of light receptor in their retinas. To them, the vibrant coral reef exists in shades of monochrome gray. How does a colorblind animal match colors it cannot even see? The secret lies in two alternative sensory mechanisms. First, their unique W-shaped pupil acts as a polarization filter. It allows them to detect the orientation of light waves, a detail invisible to humans. Second, researchers propose they exploit chromatic aberration, focusing different wavelengths at different depths in the eye. By scanning the focal depth, the brain can decode color without color receptors. They use this polarization vision to read patterns on other cephalopods, while staying hidden from fish. It is a private communication channel that operates in parallel to their camouflage. This is Insight 6. Matger and Hanlon proved that cuttlefish use polarization vision for both camouflage matching and private signaling. This complex sensory apparatus feeds directly into their learning and memory systems. Remember, the vertical lobe sits right next to the motor controls in the brain atlas. This layout allows them to remember and adapt their camouflage over time. Cuttlefish show episodic-like memory. They remember what food they ate, where, and how long ago. In lab tests, they can learn to wait for their favorite food, showing self-control. They pass the famous marshmallow test, a task once thought unique to primates. This memory allows them to recall which camouflage patterns worked in specific spots before. This is Insight 7. Josette Alves and Schnell demonstrated episodic-like memory and self-control in cuttlefish, driven by vertical lobe learning. This learning ability takes cuttlefish camouflage out of the realm of simple reflex. It proves that camouflage is a learned, memory-backed problem-solving strategy. Even more surprising, some of the light detection happens in the skin itself, bypassing the brain. Opsin proteins in the skin respond directly to ambient light, helping adjust baseline shading. This means some of the color decisions are processed locally, in the skin itself. This skin-based photoreception is documented by Ramirez and Oakley in the Journal of Experimental Biology. It shows that the cuttlefish body is not just a canvas, it is a distributed computer. When we look at the cuttlefish, we must abandon our old assumptions. This is not an automatic, reflex-driven creature reacting blindly to its environment. It is a complex problem-solving animal running multivariable computations. Its skin is a dynamic output of a continuous neural negotiation, a negotiation between its internal physiology and the surrounding reef statistics, a live debate run across 32 functional brain lobes every time it decides to hide. Every time you watch a cuttlefish vanish in milliseconds, you see the output of a 32-lobe neural debate. It is not a reflex, it is active problem-solving. If this is the kind of mind you want more of, subscribe. New episode every Wednesday and Saturday. Next time, the morning squid that runs 2 different conversations at once and decides exactly when to lie.